Hello and welcome to Preprints in Motion. Join us as we sit down with early career researchers and discuss their latest preprint and find out about their journey through the muddy marshes of academia. But we don't stop there. Every month we'll be bringing you special episodes with open science leaders where we discuss how to fix academia. Easy, right? So hit that subscribe button, leave a rating, or find us on Twitter at MotionPod. But for now, let's get into the show. Today we chat to Sonia Solar about mini brains, or more accurately, organoids as 3D brain models. I have never worked with organoids or anything even like it. Primarily do a lot of microscopy stuff, so we can chat a lot about that part.
Nice, yeah.
But yeah, organoids, we used to have lab meetings with a lab who worked with organoids, and they always had really cool images that I was always very jealous of.
Yes, the pictures we get are pretty, that's for sure.
They're definitely really, really pretty. Like some of the ones that we see in our lab as well are like, it just looks so nice where you see all the different cell types going throughout.
Interacting and
why don't mine look like that? Why don't my cells look as pretty?
Yeah, it's funny because sometimes it really doesn't mean anything. It's just a characterization or a routine step that we do, but the image is pretty, yes.
Yeah.
So I was going to start with a bit about how you've just submitted and ask about how writing was and if you're Viva ready and all that kind of stuff. But Emma jumped in and did that before we hit record. So we'll start with congratulations on getting submitted.
Thank you.
Good luck for what will almost certainly be a really nice, simple Viva, because what we're about to talk about is some really cool work. And it looks like you've kind of, when I was reading it, every question I had you kind of answered in the next figure. So it looks like you've got what's going to be a really, really good paper. So I think congratulations all round are in order.
Thanks, thank you. Luckily for me, I had some time to write my thesis. Like I have some colleagues that had to rush a lot with the last experiments. And then, in the end, you have like a couple of months to write, which can be then a very stressful process.
I wrote mine in 6 weeks. It was...
Oh my God, yeah. That's what I'm talking about.
You wrote your thesis in 6 weeks?
Yeah.
Oh my God. I took like 3 months.
Yeah, me too.
Yeah.
And did you manage well? Was your brain in shape afterwards?
The reason I had to do it so quickly is I had a job lined up. I definitely wasn't ready to start that new job. It was a job in a brand new lab, so it was setting up the lab as well, and definitely should have taken a bit of a break between those two things, but just didn't have the time.
Yeah, no, for me it worked well. Of course, it was stressful because you always have this anxiety behind, but it worked well for me.
And you also speak a whole bunch of languages, 5 of which you say you speak fluently. So did you just chop and change? It was one paragraph in one language and then jump to the next paragraph and write it in a different language, just to keep the examiners on their toes?
No, I wouldn't do that to the examiners, to be honest. It would be a bit mean. No, I have 3 languages in the acknowledgements.
Oh.
Yeah, because I'm from Barcelona, so I already speak 2 languages basically in there. Pretty much everyone speaks Catalan and Spanish fluently. And then, of course, you have English. That is the must always. And also I live in a very multilingual country. Now I'm living in Luxembourg. And so in here, they speak a whole bunch of languages. They speak German, they speak French, they speak Luxembourgish. So I speak also French, and I'm learning Luxembourgish now. And I also speak Portuguese. Very random, but I also do.
Well, I'm impressed. I speak English and not great.
No, but in the end, people that speak multiple language, what I say always is that in the end, I don't speak anything well. Like not even my mother tongue. I start making weird mistakes, so it's funny. Yeah. So
I guess a good sensible starting point would be for you to give us a little overview of what it is we're going to be talking about today.
Yes. So this preprint that you can find now in bioRxiv that I am very happy about is basically a very big part of my PhD. And in my PhD, basically, I worked on trying to increase the complexity of midbrain organoid systems. So basically, I guess you're familiar with organoid models. Well, you, Johnny, said that not so much, but basically, I work in a research center that is pretty much focused on Parkinson's disease, and everyone uses their stem cell models to to study Parkinson's, mostly genetic forms of Parkinson's disease. And in the group where I'm working, we work with midbrain specific organoids from healthy individuals or Parkinson's disease patients, so iPSC derived. And basically, these midbrain organoids, we can consider them like as mini brains from these patients or healthy people, specifically the midbrain part, which is the most affected part by Parkinson's disease. So my PhD thesis, and especially the work that I carried out for this manuscript, was about incorporating microglia in the system, because these midbrain organoids, so their starting cell population is a neural stem cell population that is already kind of pre-patterned to have a midbrain identity. So it's basically ectodermal. And microglia is mesoderm derived, so it doesn't raise in the system innately. There are some very interesting papers, especially I remember one from Ormel, where they use iPSC derived cerebral organoids, and they show that microglia in there innately develop within the system, which is pretty cool, because then you don't have to go through all the incorporating process. But in our case, because it's what is called a guided differentiation, so we want a really specific region of the brain, this is not possible. So we have to start by already a pre-patterned cellular population. And therefore we have this issue that microglia don't innately differentiate within the system. So the entire story of this preprint is that the incorporation of microglia and what happens once these midbrain organoids have microglia in there, what are the differences, especially from a gene expression point of view.
So for the benefit of folk like me, could you explain what an organoid is? And you've also used the term assembloid in your preprint.
Yes.
And maybe also just cover briefly what microglia are. I know what they are, but some people might not.
Yes. I feel a bit, threatened by an immunologist, because I don't know so much about immunology, but yes.
Neither do I, it's okay.
Okay, that's nice. So yes, an organoid system is basically a cell culture model that tries to mimic an organ, basically, like talking very simply. So in our case, we use midbrain organoids. Midbrain is a part of the brainstem, that is an area of the brain located more or less in the center, anatomically speaking. So basically what we do is we have iPSCs that are reprogrammed from skin cells, from fibroblasts, from people, patients and healthy individuals. And basically these iPSCs, we transform them, we differentiate them into these neural precursor cells. So we have a cell line in a 6-well plate there that we detach, and we put in a ultra low attachment U-shape, U-bottom 96-well plate. So it forms a ball that normally is called a spheroid, because it has this spherical shape. And these normally, when these spheroids increase the complexity, they self-organize in there, and you can see kind of a bit more complex structures, differentiated cell types, etc. You can call that an organoid. And we specifically do that to get midbrain organoids that we characterize by the expression of midbrain specific markers, midbrain neurons, especially dopaminergic, that are the ones affected in Parkinson's disease. So in our case, that's our organoids. And then, of course, like, science is advancing super quickly, and it's very cool because now a lot of people are working with what is called assembloids, right? And assembloids normally are a fusion of organoid systems. So basically you can fuse two organoids, or you can co-culture organoids with other cell types. So it increases the complexity of the system. So some people talk about the multi-lineage assembloids. So you have ectodermal and endodermal there, cell types there, or mesodermal in case of microglia. Also, there are some cool works with enteric neurons, actually, in connection with brain organoids to study the brain-gut axis. So these technologies are advancing super quickly into assembloids, multi-organoid systems, assembloid on chip, organoid on chip, and these things are super cool because you can really study interactions between cell types and systems. And in the end, you can even have what is called almost a human on chip, right? So you have like multi-organ connected with different cell types. And I think it's super interesting. It's super cool.
I mean, the benefit of these systems, of course, is that it gets you much closer to what is really going on than sort of traditional cell culture does, but without needing to use a mouse or a human. It's hard to use humans. It's not allowed to use humans. It's hard to use humans.
No.
So I guess a logical next step would be to have, these are still things that are ultimately contained in a sort of culture medium and in a dish. So, I mean, the chip aspect is probably answering this question, but have people started to do these things where you've got organoids, but you've got blood flow with them, or you're mimicking blood flow and that kind of thing? Are we getting to that level of complexity?
Yes, I think we are. Actually, it's funny because the other part of my PhD is about trying to incorporate vasculature into the organoids and trying to get somehow not blood, but medium flow in there, like perfusing them. This is still not achieved, but I've also read quite a bit on that. And indeed, as you said, the chip systems are pretty much the answers. In here, I see a lot of synergies between tissue engineering and cellular biology, because there are a lot of papers and preprints about engineered scaffolds that scientists use to grow the organoids on or in somehow, and these scaffolds are sometimes perfusible, and then you can basically pump in the medium or whatever, the culture medium in there to kind of mimic a vasculature system. So in my case, I'm trying to make assembloids actually with blood vessel organoids and midbrain organoids with microglia, and trying to get in the end a system with microglia, with blood vessels, and try to get these blood vessels closed and luckily perfusible. So you have there a system pretty much complete about the human midbrain. But yeah, a lot of things are going on with tissue engineering and with hydrogels, with scaffolds, and it's super cool what they do.
I mean, it sounds super complex. Just thinking of cell culture, it can be so difficult to culture two different cells together, never mind multiplying, organoids together. So, I mean, what is your day-to-day like? How is that? Because it's also quite a long, and Emma will speak to this, it's quite a long protocol. So, if something goes wrong, you basically have a month to kind of restart and get things up again. Whereas, I mean, my PhD was Drosophilus, so anything went wrong, it was a day. It was fine.
Oh, that's so nice.
Right. But it's one of those, you've got to really commit to an experiment, and that must be quite a stressful thing to do day to day, I imagine.
Yes, it is. It is stressful, and it's also, you have always a lot of pressure, and also because you're doing your PhD or you're a postdoc, you have always pressure on you, and you need to get results, and especially because it's normally a very long protocol. Sometimes you work quick. It has happened to me, and I don't know, you put the wrong supplement on the medium and it's 2 months that go to the bin, and it's frustrating. In our case, if you start with the iPSCs and then you have to derive the what we call the neuroepithelial stem cells, and then generate the organoids, and then co-culture them with the microglia, the entire process can be easily 2.5 months. And because I analyzed my data in a kind of an early time point, because I could go for longer. Actually, it's one of the weaknesses of the preprint. I think that if we increased a little bit the duration of the culture, we could also get a considerable amount of astrocytes, and then kind of study the interactions between microglia and astrocytes. And this, I think we should go to, instead of 35, like 45, 50, 60 days of differentiation, which means like already 3.5 months entire process.
You've clearly not learned your lesson then, have you?
No, it's been, I don't know. I always try to make it shorter, and in the end, I should have made it longer. I don't know. It's frustrating, but in the end, the result is worth it, I think.
Does Emma agree with that?
Yeah, if you get the good result. I had a bit of trouble with my enteric neuron protocol, so that was going up to like 40 days-ish, but sometimes the results weren't expressing exactly what I wanted them to, and then you're just like, cool, I'll optimize this, and it's another 40 days until you can get a result. So it's just staggering it.
I should give you the contact of one of my colleagues. She's working on that. She may, she may help you. Yes, it's a project ongoing about connecting the midbrain organoids with intestinal organoids through a channel with enteric neurons. So it's pretty cool. And I don't know, I actually don't know what's the status of that, but I could give you the contact. So she would maybe help you with that.
Yeah, no, that would be great.
Emma gets connections out of every single episode. Every single one.
I think it's just because my lab has, like, Manos likes to have his finger in a lot of pies. He likes to do a lot of things that are new and like up and coming. So we're like a CRISPR screening lab, but we also do organoids, and we do iPSC differentiations and screens in iPSCs, and then we do things like sequencing and stuff as well, like new that I don't understand.
That's awesome.
Loads of stuff. He likes to do all the new stuff basically, which is great, but a bit stressful sometimes.
Yes,
you're the one figuring it out.
So when it comes to disease models, you're looking at Parkinson's disease, but it seems to be, just a cursory glance at literature, that a lot of the organoid research, at least in terms of neurodegenerative diseases, is focused more on Parkinson's. Do we have organoids for other neurodegenerative diseases? Why is there such a focus on Parkinson's?
Well, the thing is, the research center is pretty much centered on it's focused on Parkinson's disease. So all the groups in there are working on Parkinson's disease, and well, it's important. There are a lot of works going on Alzheimer's disease, which is the first neurodegenerative disease in the world, and then followed by Parkinson's. And I think Alzheimer's disease, you can find much more things, I think, but I haven't seen a lot of organoid systems to study Parkinson's. And I think this can be because dopaminergic neurons can be tricky. So I think what I have read and what I have heard is the easiest type of neuron to differentiate and to handle and to get in an organoid is kind of a cortical neuron. Also to co-culture with microglia, they are happily co-living in the dish, and they are very happy together. Dopaminergic neurons can be tricky to differentiate, and also in the case of co-culture with microglia, it's very tricky. And this, I think, it's one of the highlights of this preprint, and it's the fact that we worked a lot to get a culture medium that allows for the survival of both dopaminergic neurons and microglia, because at the beginning it was really hard, and just the medium optimization is 1 year of work. In the end, I think it's pretty cool that we use the midbrain organoid system for Parkinson's disease, because you can really see what's happening to the dopaminergic neurons. In our group, we are also trying to advance by obtaining assembloids by fusion of midbrain with striatum organoids, and this is because normally the cell bodies in the midbrain from dopaminergic neurons project their axons to the striatum. So you also get the axons from dopaminergic neurons in the striatum. So in there you can study what is called the nigrostriatal pathway. So the entire neurodegeneration of dopaminergic neurons in Parkinson's disease could be modeled there. So I think it's super cool that we can get to an advanced model and more accurately modeling what's happening in there, also with astrocytes, oligodendrocytes, microglia now, ideally one day with vasculature, endothelial cells. So yeah, I think it's an advantage that we have this model for Parkinson's.
And how do you induce Parkinson's disease in the organoid? Because presumably the organoid starts off as a healthy situation.
Not necessarily. So what we are doing is we're going for the easy way and studying genetic Parkinson's disease. Okay. So we use stem cells from patients that have mutations that cause Parkinson's disease or increase the risk to develop Parkinson's disease. We have also engineered lines that are wild type, so healthy lines where a mutation has been introduced, a causative mutation of Parkinson's disease has been introduced, and then we basically study genetic Parkinson's disease. However, in a very small portion, like just a very small subset of our group has studied toxin exposure to try to mimic somehow idiopathic or environmentally caused Parkinson's disease, which is actually kind of the 80% of the cases. But since it's so difficult to control, we mainly focus on the genetic cases, yes.
And you're adding microglia to these organoids. Is this the same proportion you would find physiologically, or are you adding more microglia to sort of amplify the effects that they have?
Yes, so that's a very good question. I'm adding a crazy number of macrophage precursors into the system. That is true. We tried different proportions, and at the beginning, if you add too few cells, they don't survive in the system because you always have a certain degree of cell death. In the end, what we get is, so we get around 6.4, I think it was, % of microglia into the system, which is not mimicking 100% what's happening in the embryonic brain, because in the end our midbrain organoids are kind of mimicking the embryonic midbrain. So we basically our hypothesis is that Parkinson's disease has something going on already during development. So there's something in development already of people that have mutations causing Parkinson's disease. So basically we get this proportion that increases throughout the development of the brain, and in the end in the adult brain is around 10%. Even though there are different papers and different studies saying different proportions, but basically I trust the immunohistochemistry based study that counted around 10%. So it's more kind of mimicking what happens in the adult brain, not so much in the embryonic brain, but this doesn't worry me too much because the proportion can be always adapted.
Yeah.
So if you want to go for more, you would add more. If you want to go for less, you would add less precursor cells.
And what about the microglial phenotype? Because I'm assuming that would change in Parkinson's disease, and that's probably part of what causes Parkinson's disease. So how are you controlling for that in your experiments?
Yes, that is actually one of the limitations of our work for now, because iPSC-derived microglia are per se a bit activated, just because I think because of the differentiation process, the derivation and the handling in the lab, in the cell culture, it already makes them a bit stressed. So their phenotype is not completely resting. And you can also see that in the images. If you look for papers that work with iPSC-derived microglia, this own preprint that we're talking about, they don't look all of them perfectly ramified and resting and just chilling in the system, even though I only used wild type lines. So probably if I used a mutant line or a PD, a Parkinson's disease line, I haven't done it. So I don't know if I would see such a difference. So now something that we're working on is trying to get them a bit more resting. So we're trying to get the phenotype into a bit more physiological phenotype. And actually, this is something very cool that we are seeing with the vasculature part. Actually, when you have endothelial cells in the system, they look happier. Microglia look more resting. So yes, it's one of the things that should be assessed still.
Has anybody looked at microglial subtypes in Parkinson's? Just out of interest.
Yes, actually, there are a lot of studies talking about reactive microgliosis in PD. Basically, microglia get a pro-inflammatory status. They release a bunch of cytokine chemokines, and they actually further advance the neurodegeneration. So they release a bunch of stuff that damages the dopaminergic neurons, and actually, I don't know if I even read some work about targeting microglia as a drug target to try to reduce neuroinflammation and therefore trying to reduce neurodegeneration. So this is something, yeah, it has been studied, and it is true that we have a neuroinflammation caused, of course, by microglia and astrocytes in PD, and it's a pro-inflammatory phenotype.
It's the immune system everywhere. It's clearly the system we should all be working on.
Of course. It is. It's so important.
So Emma presented this at Journal Club, and one of the questions they came up with, we can ask the author. So you did a lot of sequencing in this study, but you used nuclei rather than whole cells. So where was that choice? Why did you make that choice?
So the sequencing part, I have to say, don't be too harsh on the sequencing part, because it was not me, the one who analyzed. But yes, so basically, of course, we have a lot of mitochondrial genetic material, ribosomal genes, etc., that are sometimes not what we want to see. And we have also done single cell sequencing. Like, I have colleagues, and they have seen like some, interference of mitochondrial information, ribosomal information, that in the end made the data not so clean. It was more difficult to distinguish what was what, to distinguish phenotypes, to distinguish pathways, etc. So in the end, because of previous experiences, we saw that going for nuclear sequencing was much cleaner and allowed us to get much more reliable information of what we are getting in the end.
There you go, Emma. You can report back.
I can report back to my lab. that wasn't the point of asking that question. I was just generally interested because we do single cell RNA-seq, but we do like whole cell single cell RNA-seq, and I just was wondering why the change in difference. But yeah, it makes sense if you've got previous data that suggests there was a contamination, or it not changed, or like kind of made it harder to interpret the results. Then.
Yes, it's like this. Don't ask me about the specifics because
oh no, I'm the same. We've got a bioinformatician. I don't do that.
Yes, exactly, same thing. But I know it's because of that, and in the end, it's basically much more easier to interpret the data.
So you showed in that sequencing data that there were a number of genes downregulated involved with oxidative stress.
Yes.
What's your theory as to why they were downregulated?
So we have a hypothesis in there, and it's all hypothetical because we will work on further validations with molecular assays, etc. But basically, we have seen. So these microglia that we incorporate, we have also differentiated in 2D, and we saw that they phagocytose thymosin particles. So they have phagocytosis ability. They are functional from that point of view, and we have also seen then that phagocytosis-related genes are expressed in microglia in organoids, so especially in the microglia cluster. So that made us think that microglia in the 3D system are actually functional as well. And then we have also seen that midbrain organoids with microglia, what we call the assembloids, are smaller in size. And also we have an image analysis pipeline to detect pyknotic nuclei to compare basically the amount of pyknotic nuclei in organoids with microglia and without microglia. And we saw that they are less in the organoids with microglia, at least that's what the pipeline detected. So this, together with the gene expression data that shows downregulation of oxidative stress-related genes, made us think that maybe microglia within assembloids are able to somehow phagocytose some metabolic waste products, maybe even some cellular debris and some stuff that in the end is causing oxidative stress in the system, and in the end is leading to a more, let's say, a healthier status in organoids when microglia are there than compared to the system without microglia. So we think this is the story that may be happening. Of course, a confirmation should be done, but it's what we speculate.
Why were your assembloids smaller than the organoids?
So we think it could be. We have really no clear answer, but we think it could also be because of this, because if microglia are phagocytosing cell debris, the core of the organoid that is very rich in dead cells basically may be smaller, and in the end the organoid itself may be smaller because of this. It can also be something else that microglia are doing, because in the end the culture media in both cases is the same. So we are culturing in the same conditions for the same amount of time with the same medium, and the only difference is that one has microglia, the other doesn't. So this is one of our hypotheses. Of course it should be confirmed, but it's the same. It's also matching the fact that this pyknotic nuclei amount is lower, so it could be maybe because of that. We are not sure.
I mean, that's a fairly simple thing to test too, I guess.
Yes.
So what are your plans with the assembloids? What is it you want to look into further with these?
So now I have a colleague, a postdoc in my group that is already applying this model to study genetic Parkinson's disease in LRRK2. So it's a gene that when it's mutated, it causes Parkinson's disease. And basically he's focusing on neuroinflammation, also seeing if, for instance, organoids, he will look at that in the future. Maybe diseased organoids with healthy microglia have milder phenotype than diseased organoids with diseased microglia, or wild-type organoids with diseased microglia are worse than just wild-type organoids with wild-type microglia. And basically to target what are exactly microglia doing in there, how are they contributing to the disease, and what is happening with neuroinflammation in the system.
Can you take microglia from patients and use them in your system directly?
You mean non-iPSC derived? Do you think that is possible, obtaining primary microglia from humans?
I was just about to say, Johnny, do you know a way that you can do that?
Yes,
well, no, I don't know.
I have seen studies with the primary mouse microglia in co-culture with human neurons or mouse neurons. But to be honest, I don't know how could you get human primary microglia without causing...
Could you differentiate macrophages into microglia?
Why would you do that if you could just get microglia, the differentiate?
Well, I'm thinking of if you take them directly from a diseased patient,
You could take the iPSC, you could take the skin cells, make them iPSCs with disease, and then go to microglia.
Yeah, but there just might be a difference in those more, I don't want to say more natural, but the sort
not
of
reprogrammed,
already differentiated microglia.
Yes.
Compared to the ones you're differentiating.
That's for sure. If you would use primary microglia, it's a complete different picture. They look much more resting. It's much more close to the physiological situation, if you like. That's the limitation of iPSC-derived microglia, yes.
I mean, I wasn't volunteering to give my
I thought you had like a technique you knew about or something. And I'm like, oh, that's interesting.
No, I'm just talking.
I would be interested, yes.
So one of the really cool things about this preprint is that you did all the sequencing, and that's where most people would have stopped. But you didn't stop there. You then went on to do functional analyses, which I always like to see. So could you talk a little bit about some of the functional assays you did and what they showed?
Yes. So some of the stuff we checked was the electrophysiological properties of the neurons in the system with and without microglia. And basically we had a collaboration where they did patch clamp, single cell patch clamp in the organoids. And then we did a multi-electrode array assay in our lab. And basically both assays showed more or less the same. And it was in both cases just to test the spontaneous activity. So we didn't stimulate the system. And basically what we saw is that the spontaneous activity of neurons in the case of the organoids with microglia was somehow higher. So basically we saw a lower threshold to fire an action potential in neurons from organoids with microglia. And basically this more spontaneous activity is somehow associated, and it's normally related to a higher neuronal maturation. This we link with the fact that we have upregulated genes related to action potential, for example, and from the single nuclei RNA-seq data that we get. And so basically we put all this together, speculating that neurons may be more mature in the presence of microglia. Of course, some more things could and probably will be done, like checking spine morphology in the dopaminergic neurons with microglia to see if they are more complex. Also check specific maturity marker, protein expression, and stuff like this. But it was an insight into what is happening to maturation in there. And I think it was pretty cool to see that as well.
Well, you're making me want to work with organoids now. So
you can come with us. You're welcome.
So we should probably move on to the more general preprinty questions we ask. So we like to talk just general experiences in academia with everyone. So one of the things we ask everyone, because it's kind of part of our remit of doing this, but also it's interesting to see why people do it, which is whose decision was it to preprint this and where did that decision come from?
So the decision was basically, I don't know if it was my PI first who suggested it, or I think it's a normal practice that we do in my group. The thing is, this paper, this preprint was ready a bit before it was out in bioRxiv, but it was also linked to a patent, so we couldn't share it for the moment. And then, once the patent was like, all the documents were perfectly fine and everything was done, the first thing my PI said was, let's put it in bioRxiv so it's available. And I am very happy about it. And I think it's how things should done, because the publishing process can be interesting.
A nightmare.
Yes. So I think it's so important that preprinting and making your data available for everyone, so people can also use it and use it for making their research better, or maybe stop using the approach they are using and going for something different, contacting me, contacting the first author, the last author, and ask for some support. I think it's how it should be done.
That's what we want to hear.
Yes.
So you're still early in your academic career, if that's what you're going to have. But you've already seemed to have jumped across fields a little bit. So you've got some neurobiology, some cardiovascular biology. How do you find that experience? So this is something I've done at sort of every career step. I seem to jump and do. I'm still in immunology, but I seem to work with different immune cells, different part of the immune system, change up different organisms. How do you find doing that? Is that, I mean, in terms of this project, it looks like it's been very beneficial.
Yes, I did that. To be honest, I don't know if that's how most of the people think, but I really like research and I like science, and I am a biologist by training, and I like biomedicine in general. So I love the topic of my PhD. I'm amazed by it, and I really like neurobiology, but I also enjoyed working in microbiology as well. I enjoyed working in cardiovascular diseases. So I think it also enriches so much your academic career, and you can also learn so many different techniques that you can then apply to the other fields somehow. And I think also in your case, if you're working in different fields of immunology, for sure, it can be only beneficial.
That's what I keep saying. But
yes, for me, it is. And I don't regret at all having jumped like this. And I don't know, even if at some point in my past, they would have offered me an internship in plant physiology, probably I would have also gone for that, because I find all of it so interesting.
I have limits.
No, I'm very curious. And now that I am in neurobiology and I like it so much, and I have seen so much in depth, because of course in the past it was internships and Erasmus, the master's internship. But now that I've seen this topic really in depth, I think I would like to stay here. But I don't regret having jumped like this. I am really happy that I did that.
Yeah, I think more people should do that.
Yeah.
So what are your future? You've just handed in, so you obviously immediate plans are Viva, which based on this conversation you're more than prepared for. But what are your longer term plans? Are you planning to stay in academia, or are you going to jump ship at the first opportunity you get, which is the small thing to do, really?
Yeah, a lot of people think like that. To be honest, for now, once I defend my thesis, I consider myself very lucky, because I will stay 1 year as a postdoc in the group. So I can continue my experiments. I can finish some stuff that is unfinished. And I'm very happy about it, because we have some interesting things that I want to continue. And luckily we can get maybe another preprint about the other part of my PhD. And so for now, that's what I will do. To be honest, I was so stressed and so busy with the PhD that I have not thought further. So I have no idea what I will do. Academia is, I think it's very hard. So I don't know if I will forever, like forever is a very big word, right? But in the long term, stay in academia. But I am really open to also exploring new career paths, right? So I like academia and I like research, but I am not close to other ways.
I think it's probably the best way to do it. Every day I think about leaving academia or staying in academia. Depends how the day's been.
It can be hard, I think, especially the kind of contracts and instability, insecurity.
So that's what I was about to get into, which is, one of the things you focused on as one of the negatives is the lack of job stability. Yes. And I think this comes up quite a lot here. And I think we've asked somebody this before, but what would be the ideal system in your mind? If you were to design academia, what would you do to change that lack of stability?
Big question.
Yes, I don't know.
On the spot as well.
I don't know if I have I could say stuff, but then probably someone that knows about the system and how it's structured and about the big papers and stuff.
I know all this, so you speak freely. If it sounds terrible, we'll just edit it out. It's all right. It's a safe space.
I don't know. Maybe somehow because it's sometimes easy for it's easier for example, technicians to get permanent positions in a group and work during a long period of time in different projects for that group. So maybe an option would also be like trying to do something like this for postdocs, so instead of being linked to a project that is 2 years, 1 year, 3 years, trying to somehow with HR from whoever you are, trying to say, okay, I am a kind of a permanent postdoc in that group, and now I cannot be fired without getting a compensation somehow, and I will work in different projects, and I will keep being enthusiastic about it. I think it shouldn't be so difficult, right? Like, is it crazy what I'm saying?
No, it shouldn't be. I have heard of some professors who, when they negotiate their contracts, they stipulate that they want a postdoc, not on a grant, but as part of their contract. So as long as they are around, they have the money for a postdoc. And so in theory, you've got a permanent postdoc. One of the things that comes up is the idea that you could have sort of roaming postdocs. So a research institute might pay the salaries of a handful of postdocs, and they kind of cover maternity leave, or they'll jump on and cover sick leave and things like that, so that they've got a job all year and they're just jumping around different research groups.
Oh.
I mean, I think having a postdoc career pathway is desperately needed, because it's the one thing that there's so many people who don't want to be a PI and just love being at the bench and want to be a postdoc forever, but it's so difficult to do that.
Yes, it is crazy. And something like this, like a jumping postdoc in a research center, that's also nice if you like the topic of the overall topic of the research center. I just think it's crazy, right? Like being in a 2-year contract. Imagine that, I don't know, just hypothetically, you are 38 and you're a woman and you want to get pregnant and have a baby, and then you have a 2-year contract, and then you have a pregnancy in between, a labor in between, and like it's of course it's hard for men, but I imagine also this situation.
Not that hard for us, really, honestly. we have such an easy ride compared to pretty much anyone else in academia, and it's really not fair. It's not how it should be at all.
No, I think it discourages people, and I think there are so many valid and super intelligent people that just leave academia when they love academia, and just because they cannot handle that anymore, and they just want a normal life with a normal job and a normal salary.
Normal salary.
Yes,
it'd just be nice not to be like constantly chasing something, like we've got to publish in our PhD after 3 years, we've got to publish from our first postdoc, and if we don't, it's not like in a permanent position somewhere where if you don't hit that deadline, or you're just working normal 9 to 5, you're not going to lose your job and not be able to find another one. I mean, Exactly. If you do really badly, then maybe, but not if you've done your job well and you just so happen to be unlucky and things can't get published in time. It's really difficult. And then add the whole like having kids and stuff on top of that, stresses me out quite a bit.
Yeah, me too.
But to end on a positive and not a negative, because I had to have a think there to figure out how to switch that one back around. One of the things you're really excited about in science is the creativity and just where it can go aspect.
Yes. So I love painting. I try to be creative. I try to be artistic, and somehow I compare it with what you can do in academic research. So in our case, of course, it's not always the same. It depends on the group. It depends on how you organize, how your PI react. But in my case, I was quite lucky because I could always choose which way to take, which way to go for my experiments. Also, I work in somehow abstract project, like there is a midbrain organoid, just try to optimize it, put microglia in there. How, I don't know, but just try to put it in there. And not only in my case, but there are other projects with different models and also molecular based projects, I guess, that are similar, right? Like just try to get this, how, I don't know, just somehow be creative, right? Which is kind of, there's a bit of irony in there because we are meticulous, we are scientists, we are logic, but somehow we also have to be a bit creative.
I think people forget that. So I've been doing this thing online where school kids ask you questions. I did it all last month. Some of it was really intense. And one of the things they ask is like, what subject should we do other than science? And those kind of things. And I always come back to the creative stuff, because things like writing skills are obviously really beneficial, but also just those things that make you think differently, because so much of science is troubleshooting, and troubleshooting is nothing more than thinking differently. It's coming at the same problem as many different ways as you could think of doing, and people do often forget that, I think.
Yeah, totally. They imagine us somehow as robots that just go one way and cannot imagine and then have some creativity. And it's something that I actually learned in my PhD, I think, because at the beginning, you're always scared to ask, and what if we did that? What if we went that way? And in the end, I am like, why not getting microglia from a human person? Let's just the same stuff, right? No, really, like now I imagine and I ask, and if it doesn't work, it doesn't work. But the possibilities are pretty much endless, I think.
And that is exactly where we should end on. The possibilities are endless is a great ending.
It is.
And it's a positive. Look at that. Look at how that worked out. There
it's not all bad, of course.
It's not all bad. Some of it's bad. But a lot of it's good. Okay. Well, I enjoyed that, and I learned a lot, because I knew basically none of that going into this. So that was really good. Thank you.
Yeah, thank you so much.
Thanks for having me. It was really very nice.
Okay, and that is the show. If you enjoyed listening, then hit that subscribe button for more and leave us a review on whatever platform it is you're listening on. You can reach out to us on Twitter at MotionPod or online at preprintsandmotion.com. Didn't enjoy that? Well, we're all scientists here, so send us your review and let us know what works or what you'd like to hear more of, or less of. But until next time, have a good week. So the way we normally do this is we just jump straight in with the questions, and I will do all the intro stuff separately when John, who's not here, prompts me to do it because he's about to finish the edit off and I still haven't done it.
Okay.
We're a very, very well-oiled machine over here. We get everything done on time and it's great. It's so much smoother than we up here.
But the result is awesome. I have listened to some of your podcasts and it really looks super well structured and I like all of them, the ones that I've listened to.
See, that's what we like to hear. We need to get a soundbite of all the guests saying nice things about us as an episode.
We can
clip that. We can clip that soundbite. That would be good.
I'm being totally honest. I really like it.
Good.
It's so nice to hear.
That's what we're here for.
Yes.
Where do I find out about the different bioarchive licenses? This CC, BY, CDXY nonsense is driving me nuts.
ASAP Bio have a resource for that.
Ugh, that's your answer for everything.
That's because they have everything you need to know about preprints.
Sure, they probably have the basics, like info on the preprint servers, but what else is there?
There's so much more. Looking to post a preprint but not sure what different journal policies are? They have a collection to help you out with that. There are meetings around preprints and associated services. If you want to know how preprint adoption has changed over time, there's even a page on that.
And COVID?
They have a big section on preprints and the pandemic, plus some really cool infographics for communicating preprints.
And university policies? Surely they don't have that.
They collect uni policies where possible.
Okay, they do sound pretty impressive, but is it not a bit of an echo chamber?
It can be, but ASAP Bio also engage with people who don't love preprints and have concerns. So we had an excellent discussion on this very topic a couple of months ago.
Oh, is there anything ASAP Bio don't do?
Honestly, no, they're so nice over there. They were so quick to jump in and support this show. It's your one-stop shop for info on preprints and open science initiatives, so head over to asapbio.org to learn more and subscribe to their newsletter for the latest in preprint news. If you want a deeper dive into the world of preprints, then look out for the next recruitment of ASAP Bio Fellows.